Okay, we're going to start by talking about just a general overview of microbiology, hitting some topics. And let's begin by first talking about some case studies to try to explain the importance of microbes and how they infect us. So first up is Ivan Goes to Chicago, and this is going to be a story about drug-resistant TB, or this can also be about COVID-19, anything contagious, highly contagious. And so Ivan lives in Russia, and he wants to visit his family who lives in Chicago. And so at time 0, Ivan is going to board a plane and go to New York City. And this is about 10 hours. And on the plane, there's 256 passengers. And then Ivan is going to continue his journey to Chicago. And on the Chicago plane, there's about 210 passengers. And this is approximately 3 hours. Now in Chicago, Ivan goes to visit his family members. And then he gets sick and is admitted to the hospital. For respiratory disease. So now if he has active TB or COVID, you're going to have to do contact tracing, and this is going to have to go back into identify who Ivan potentially could have exposed to this particular pathogen. So in Chicago, we had 210 people on the plane. Ivan, of course, went to visit his family, so that leaves 209 people. And some of them went to Chicago, stayed in Chicago. Oops. And then some transferred. And in New York, you had 256 passengers. So we have Ivan, who went on to Chicago, so 255 passengers. Some stayed in New York, and others transferred. So very quickly you can see how a contagious infectious disease can spread worldwide because of our transport system nowadays. Next up is influenza. Influenza is probably one of the most important pathogens for humans, and part of the reason is because it is a zoonotic infection, meaning that it infects animals as well as humans, so it has animal reservoirs. Pathogens that have animal reservoirs are very hard to eliminate because they can kind of hide in those reservoirs and then infect humans as their interaction becomes, the interaction increases and the virus finds another host. So influenza in general is not highly lethal. However, if it does get into susceptible populations such as those over 65 and those with underlying respiratory conditions, it can be rather lethal. So in the 1918 pandemic, you had about 50 million people worldwide die from what was called the swine flu. And in SARS-CoV-2, we have about 212 million people infected and about 4.4 million people dead. And you have to consider for a moment with this 50 million people dead by swine flu, this was over a period of about 24 months. I'm going to say 12 to 24 months, because most of the death did happen within the first year. And then with SARS-CoV-2, we're at 4.43 million, and we're almost at 24 months. So the 1918 pandemic, of course, led to higher mortality rates than what we're seeing with our SARS-CoV-2 today. They are very different viruses, though. Now, I just want to point out there is some variants of influenza that have very high mortality rates. The H5N1 influenza virus has a 60% mortality rate. However, it is not very infectious, very difficult to transmit from person to person. People who came down with this H5N1 highly high mortality rate virus typically picked up the virus from the animal reservoir themselves. And so let's just talk a little bit about the H5N1. The H5N1 arose in a very unique way, which is we don't typically see. So we do have our bird reservoir, and we do have our swine reservoir. And then, of course, we have our humans. Now, swine are very interesting. A bird flu can infect swine. A swine flu can infect birds. A swine flu can infect humans, and a human flu can infect swine. So swine is sort of the melting pot. Oops, not pot. Now, what's rare is the jump from birds to humans. And so this is rare. And this typically is more lethal. And that's what we saw with the H5N1. It was a bird flu that jumped directly to humans and skipped the swine melting pot. And so that had a more lethal outcome. Now, it's not just all about viruses. Bacteria can also cause problems. So E. coli has some pathogenic strains. One is the O157H7 as well as the O104H4. Now this O and H are referring to certain components of the E. coli that identify it as a specific strain of E. coli. So the O is going to be the outer membrane lipopolysaccharide, or the LPS, and the H refers to the type of flagella. So O157H7 just simply means it has number 157 identified LPS on its surface, and it has this number 7 flagella that was identified. And you can say the same thing for O104H4. And so E. coli, if it's the right strain in the right location, is fine. It's part of our normal flora. It's in our colon. If it gets into our small intestine, then it can be a problem. If it's a strain from cows, it can be a problem. One of the things to keep in mind when we do get food poisoning from things like undercooked meat, undercooked chicken, it's because in those species, they have in their normal microflora a pathogen for us. It's not pathogenic to them, it's part of their normal flora, but if it gets into our intestinal system, it causes an infection in us. And so that's why it's important, because we eat these types of animal products commonly, to treat them properly. So E. coli has a broad range of diseases. You can have life-threatening diseases, and these are you can have diseases of the urinary tract infections. So E. coli can disseminate into your kidneys if it gets into the urinary tract, and so it can cause severe infections of the kidneys, and it can cause acute kidney failure. You can also have non-life-threatening illnesses. You can have bloody diarrhea, dysentery is what it's called. And so this is going to be where the bacteria is actually killing your cells and the cells are sloughing off, and so blood is leaking out. So this can lead to severe abdominal cramps, watery diarrhea with blood, vomiting, and inflammation. And then you can have the pathogenesis of your cattle, O157H7 E. coli. And again, this can be from unsanitary slaughtering. And so one of the reasons why hamburger meat is often contaminated is because hamburger meat is a ground up meat. So it has increased surface area. It has increased exposure to potential contaminants, whereas a steak is a clean cut meat, and so it doesn't have that potential exposure. You can also get the bacteria in milk. So pasteurization of milk is very important, and from water sources as well. due to feces. And so it's just important to understand that your bacteria in normal things that we eat can cause problems as well. Now, our last story is that a hospital can be dangerous. And what this means is you have a grandfather who comes in for a hip replacement. And the operation goes just fine. And the grandson goes to pick him up. And when he gets to the room, there is a biohazard sign on the door and do not enter. And so what's going on here is you have nosocomial infections. Or hospital acquired. And so basically the elderly person went in to get a hip replacement, got exposed to something in the hospital. It could have been a staph infection, it could have been aspergillus, but he was in a compromised state due to his age as well as to the operation, and now he has this probably staph MRSA type infection with the biohazard sign of do not enter, and he's going to be in the hospital for a much longer period of time because of that infection. Now, of course, you can say that there's a relevance of microbe diseases to healthcare because, of course, people are going to present with these infections when they go to visit their clinics. And so just showing just a little bit of what's happening, one thing to keep in mind is there's been several leaps in protecting us from infectious diseases. Public health departments, establishment, and the cleaning of water, the cleaning of sewage, just making sure that the sanitation is increased. Chlorination of water, drop the number of bacteria that were actually in the drinking sources. We, of course, had a spike that overcame all these, and this is with the influenza pandemic. And then we start to see immunizations popping up, as well as antibiotic usages, a few more vaccines coming up, more vaccines. And you can see that in the developed country, we're able to control much of the infections that at one time were a serious concern to just people in everyday life. And so the healthcare system with respect to microbiology is always going to be there, but there are some barriers in place that help to minimize the potential for getting these pathogens in our environment. One thing to keep in mind is the most common infection is a virus. So it's not bacterial. Okay, so let's talk a little bit about infectious diseases. So there's only a tiny fraction, tiny fraction of microbes that cause disease. And of these tiny fraction of microbes, only a fraction of them are pathogenic for humans. Now, a microorganism that causes an infection is called a pathogen. And that's a pathogen. Now, some few definitions. Pathogenicity is the ability to cause disease. Whereas virulence is a measurement of disease severity. So keep in mind that even if a pathogen is able to cause a disease, it doesn't mean it's always going to be a severe disease. We get colds all the time, and they do not turn, for the most part, into a severe infection. Now, for some pathogens, that's not the case. If you get infected with Ebola, more often than not, you're going to get a severe infection. Now, another thing is that we have normal flora. And these typically are going to protect against pathogenic microbes. And this is basically by space. They take up space. They're able to live on our bodies or in our bodies much easier than a pathogen. And so by them being there, they don't allow pathogens to colonize us. However, they can become pathogenic. And this is typically when what is referred to as an opportunistic pathogen. And opportunistic pathogens typically possess a mild degree of virulence. What this means is that they really don't cause severe disease. They really don't have much that they can do to actually cause an infection. However, if the conditions are right, if you have a compromised gut, for example, inflammatory bowel disease, celiac disease, then your gut microflora can become opportunistic pathogens. We always have primary or obligate pathogens. And this just means what it is. They're always going to be associated with disease. And so most pathogens are going to be looked at from the perspective of epidemiology, pathogenesis, and host defenses. So let's just talk about epidemiology. Epidemiology is basically the study determining the frequency and distribution of disease. Okay, and so pathogens are going to be studied about how well they meet 5 requirements of infection. So pathogens, 1, must get in, so they have some sort of entry. And then they have to stay in, so they have to establish themselves. And then they have to have some sort of virulence factors. And so this is going to help to defeat, at least for a period of time, host defenses. And they're going to cause some damage. Cause some disease, and then they're going to be transmitted. Now, pathogens transmission are going to be air, such as COVID-19, food or water, such as E. coli, insect vectors, such as malaria, or person to person. Sexual transmitted diseases, for example. And then there's also going to be classifications according to where they are. So there's geographic locations. So are they going to be worldwide? Or are they going to be restricted? So some pathogens require vectors, and so the vector is going to limit where the pathogen can actually be, and that's such as malaria, dengue, and those type of infections. And then others are going to be worldwide because they can be spread from person to person. They don't need an intermediate, for example. And those are things like influenza, COVID, measles. And so all of those can be spread pretty easily. Now, the other thing about epidemiology is outbreaks. So what is causing the outbreaks? So outbreak cause can be influenced by many factors. Socioeconomic, ignorance of how infections occur, poor hygiene, and of course natural disasters. Those can all influence the epidemiology of an infectious disease. Now, pathogenesis is different. Pathogenesis is the ability to cause disease. So in typically a microbe that is pathogenic has virulence factors. And these virulence factors help the pathogen persist, cause disease, and escape defenses. So that's what these virulence factors are doing. And then bacteria pathogens are going to be very unique. They can establish themselves, for example, our bacteria pathogens have digestive enzymes. So they need to have nutrition, and so they digest our cells. And they break them down into proteins, fats, carbohydrates, and they use them. They can also produce toxins, and this can help to kill our cells as well. And again, the purpose of killing our cells by bacteria is to generate the nutrition they need in order to survive. Viruses can kill the host cell. And then oftentimes the damage is immune mediated. Now, the host defense, of course, is going to be very important in whether or not a pathogen does a lot of damage or little damage, and to the point of whether or not the immune system can control it before the individual succumbs to the infection or the response to the infection. So an infection is very complex. It's basically a competitive struggle. It's a competition between the host defense and the pathogen. So it's host defense versus pathogen. And this is very competitive. The outcome is going to be based on the success or failure of the host defense. So failure of host defense is an infection. Most microbes that you encounter are not going to cause an infection. Most microbes that you encounter, your innate or your first line of defense is able to clear it. So you have your two basic host defenses, You have one is your innate, your first line of defense. And so this is going to be things like your barriers. This is going to be your macrophages. This is your neutrophils. NK cells, complement. And these are going to control most of these. Your innate, of course, is nonspecific. It IDs patterns. And then your second is your adaptive. Now, if it gets to the adaptive, typically that means that the pathogen has been able to overcome the innate system and now is causing an infection. And so adaptive is going to be specific for the pathogen, and it's going to involve memory. So any pathogen that is able to overcome the innate response, you want to make sure your adaptive response remembers that pathogen. So when you see it again, which is very likely in your environment, your acquired response, your adaptive response will be able to eliminate it very quickly. Okay, so we also have treatments for infectious diseases. So primarily when we think of treatment, we think of antibiotics. And this is only for bacteria. They do not help you against viruses or fungal infections. There's also disinfectants. There's antiseptics. So this is for skin. These are usually for surfaces, instruments. Antibiotics are going to be toxic chemicals. And so you have to use them selectively. And of course, you want them to kill the pathogen, but not the human. So just keep in mind that there are some rules about treatment. So bacteria are easier than your fungal and viruses. But keep in mind also fungal and viruses and an intact immune response can clear. Bacteria are hard for your immune system to clear. So fungal cells are going to be interesting because they are going to have some similarities to human cells. So there are very select chemicals that are toxic to fungal elements that are not toxic to our cells. However, fungi do not mutate. And so these very old chemicals and treatments are still available today because you don't have much mutation in those categories of pathogens. Viruses are intracellular parasites, so they have to be blocked before they actually get into a cell, or all the infected host cells must be killed in order to eliminate the virus. Now, the best treatment, of course, is prevention. So prevention of food poisoning, cooking your meals properly, prevention of vaccine preventable diseases, getting a vaccine. So prevention is really public health measures as well as immunization. And viruses are easier to immunize against than bacteria. So these are our vaccines. We have more vaccines that target viruses than bacteria. Okay, just a little bit about some terminology, concepts of metabolism. Metabolism for any organism is central. It helps you to understand why a pathogen can be a pathogen and why it does what it does. Um, so, carbon, necessary for growth, for us as well as for microbes. And there are two processes which we can obtain carbon: autotrophs from inorganic substances and heterotrophs from organic molecules. Almost all infectious organisms are what we refer as chemoheterotrophs, which they can break down other organic molecules. So they're going to get inside of us. A virus is going to get inside of you, hijack your cells, use your cells, make more virus. Bacteria is going to get inside of you. If it has virulence factor, it's going to digest your cells to take all those nutrients for itself. So they're going to break down other particles. Now, just really quickly, I'm not going to spend too much time because we're going to go through all of these different viruses, but just really quickly about what the different differences between these different categories of pathogens. We have our viruses, which are the smallest infectious particles. They are true parasites. They cannot live without being inside of a cell. Regardless if it's an animal cell, a human cell, they have to live inside of the cell. Key. They are responsible for most infections. They're the most common pathogen for humans. They have genomes, RNA or DNA. There are some exceptions. We have prions, which have no nucleic acids. This is actually an infectious protein. And we'll talk about that in a future lecture. And there's also a mimivirus, which has both RNA and DNA, very unique to that particular virus class. This is just an example of a virus life cycle. Again, you don't have to memorize this. This is just giving an example of what goes on in a virus. A virus has to bind to something. It has to infect. It has to get inside the cell. It then is going to go through an uncoating process. So here we have coronavirus. It's the same thing. Coronavirus has to get inside the cell, and it has to have an uncoating process where its genome is released. Basically, two things are going to happen. Your virus is going to make more genome. So it's going to make more, one, it has to make its genome. And then two, it has to make its proteins. And then it's going to bud and make more viruses. And it's the same thing with your coronavirus. It makes more genome, it makes more protein, and then it makes more virus particles. Bacteria are a little bit different. Bacteria are self-sufficient. They have their own DNA. They have their own way to make their proteins. They can do oxidative phosphorylation using their cell membrane. They are going to be simple from the perspective of a cell. They don't have any membrane-bound organelles. So you are going to have no nuclear membrane, no Golgi, no ER. They're going to reproduce by asexual division. They have a cell wall that has gram-positive or gram-negative. Gram-positive is a thick peptidoglycan layer. Gram-negative is a very thin peptidoglycan layer. However, it has two cell walls. And so it's going to have an inner and an outer. There are also some that don't have a cell structure at all, and they're going to have to be able to live inside host cells or in some sort of environment that supports that. Bacteria also have a lot of different shapes, so do viruses. And here's just an example. They're going to be round. They're going to be sort of rod shaped. This is a rod shape. They're going to be spiral, so you can have a spirochete. Single cells, chains, clusters. So chains are going to be, for example, your streps. Clusters are going to be your staph. Next up, we have just a very quick explanation of your bacteria life cycle. So bacteria are going to grow, and so they're going to have an exponential phase in which they are growing. They're dividing and replicating. So this is antibiotic sensitive. And then they're going to have a plateau phase. This is where their replication equals nutrients. And growth is excess nutrients. And I shouldn't say replication, I'm sorry, I should say bacteria. And then you have your death phase. This is where bacteria are greater than nutrients. And this is where they are antibiotics insensitive. And then, of course, the cycle will repeat unless you eliminate the bacteria. Okay, so the bacteria have a lot of nutrients. They grow. While they're growing, they're metabolically active. That's where antibiotics can get in there and kill the bacteria. When they hit the peak is where their nutrients are diminishing, and then they go in the death phase. And in the death phase, they're antibiotic resistant because they are not metabolically active. They're not taking things up. They're trying to survive. And then the cycle will repeat. And we have our fungi. Fungi are going to be able to live outside of our bodies, of course. They are not obligate parasites. They will infect us. However, our immune system can control them very easily unless they are immune compromised. They do have more complex structures. They are going to have nuclei as well as mitochondria, Golgi-ER. They have two different forms. They have a unicellular form, which is a yeast, and then they have a filamentous form, which is a mold. Some of them are going to be dimorphic. So we have our pathogenic fungi, and they are dimorphic. These are Histoplasma, Blastomyces, and Coccidioides. And these are going to have what we call thermal dimorphism. So what that means is outside they are a mold, and then at 37 degrees inside our body they are a yeast. Now the fungal life cycle is pretty straightforward. Basically they are going to divide and replicate, and when they hit a period where they don't have sufficient nutrition, or it's a little bit of harsher environment, they are going to make spores. And so basically you have mycelium, and then you can have your plasmogamy, which is fusion of the cytoplasm, and you get a dikaryote stage. Karyogamy, fusion of the nuclei, you get a diploid stage. You go through a process of meiosis. You can have your spores form, germination of the spores, and it continues. Now the asexual is a little bit easier. You have your mycelium, they have a spore structure, produce spores, you get germination, and then you go back to the mycelium. Both asexual and sexual reproduction form spores. So you see that in both of them. And then parasites are going to be the most complex. Parasites are true eukaryotes, and they have different sizes. We have our very large tapeworms and our very small pinworms, for example. And sometimes their life cycles are very complex. So I have here an example of a pinworm. Pinworm is a very simple life cycle. And so for a pinworm, you are going to ingest the egg, and the egg is going to travel into your gut and into your small intestine, where your larvae are going to hatch. And then your larvae are going to develop into male and female worms. And then your females are going to become gravid, which means they are egg bearing. This is gravid. And then the egg-bearing females are going to move or migrate to the perianal region at night and lay eggs. And the eggs are going to be on the perianal folds, and they're going to mature within 4 to 6 hours, and then the cycle will repeat itself. So typically this is going to infect children. They are going to constantly infect themselves because they will scratch because it's very itchy, and then they'll put their hands in their mouths and they'll just keep reinfecting themselves. Now another parasite is malaria, and if you look at this life cycle, it's very complex. I'm not going to go through the entire life cycle, but just to point out a few things. Malaria starts in the human by infecting the liver, and then it goes to a blood stage. It has a liver life cycle. It has an erythrocyte life cycle, and then it's going to make gametes, and the gametes are going to be picked up by the mosquito, and then the mosquito is going to have the mosquito life cycle, and then the mosquito is going to infect humans by taking a blood meal, and you're going to inject sporozoites. So the sporozoites are infectious for humans. The gametes are what the mosquito picks up. And then you have various stages. Some of the more famous stages is the ring stage. So this is very diagnostic for malaria. You just get a blood smear and look for the ring stage. Okay, so that's just a very general overview of the microbes.